So what is biomedical research? So in general, it is a broad and interdisciplinary area of sciences that focuses on the prevention and the treatment of diseases that causes illness and death in humans and animals. So my program focuses on combining several different fields of science in order to address these kinds of problems. Fields such as immunology, which is the study of the immune system, biochemistry, which is the study of chemical processes related to living individuals, physiology, which is my focus, the study of functions in living organisms, neuroscience, which is the study of the brain, and cardiovascular science, the study of the heart. So in essence, biomedical research takes all of these fields and all of this knowledge and kind of filters it into a specific disease problem. And we use each one of these in order to address that problem. So it's essentially many fields of science working together to solve a problem. So who does biomedical research? Now, biopharmaceutical will look similar to this, but I'm going to kind of break down what a typical academic lab, which is what I'm currently in. So we first have our principal investigator. That's the tippity top of the hierarchy of the lab. They find all of the funding. They hire everybody. Their research focus is the focus of the lab, right? And so that's usually headed by a person with their Ph.D., an M.D., or an M.D. Ph.D. It's very atypical that you'll find somebody outside of that unless you're in engineering or something of that matter. So the next would be the lab manager. That's kind of like the mom of the lab. They are focused on bringing in all of the materials that we need. They're the backbone of the lab, so they set up all of the meetings and they make sure that our lovely interns and graduate students, lab technicians, and research associates are on track with their projects and can communicate their projects. So next we have our research associates. Sometimes they also are known as postdocs or postdoctoral people. They are early career scientists who are interested in either learning a new field or deepening their knowledge and experiences within their same field. So a research associate works on one project in the lab and produces data, papers, and even grants sometimes. They're kind of like a mini PI, and they're training to eventually become a PI. So next we have our laboratory technicians, the other backbone of the lab. I was a lab tech at my biopharmaceutical company, so I am especially passionate about their role. They are optimizers. So they look into the instruments in the lab. They look into the validation of certain processes in the lab. They are very important for the way that the lab works, right? So then we have our graduate and professional students. That's also me, so I'm quite passionate about that. They are usually master's students, PhD students, MD, PhD students, or MD students when they have the time. And we basically look at a smaller project. Sometimes we can develop our own funding, but we're really there just to be trained in the field. So next is our interns, our lovely, beautiful interns, usually undergraduate students or sometimes volunteers or people who just have time to work in the lab. And they are instrumental in helping to do some of the smaller tasks in the lab and kind of learning how a lab works. So you kind of start from the bottom with the intern and you work your way up to a principal investigator, which is something I hope to be someday. So how does biomedical research work? For those of us like me who have forgotten the 4th grade, we have the scientific method. Everybody remember that? I know everybody's comfortable with this, but we'll walk through it. We have our question. Then we do a little research. Then we develop a hypothesis, perform experiments, observe, analyze the results, and present the findings. Now, a biomedical researcher or any researcher will apply these methods to their research, but it gets kind of a little complicated. So I'll walk you through the scientific method and then how I apply that to my own research question. Now, to give you a little background on my research, I look at how fats that you eat are processed through your body and are delivered to the kidney, and whether or not those fats in higher concentrations from higher amounts of fatty foods can contribute to the probability of you developing kidney disease in the case that you have diabetes. Okay, I hope I didn't lose anybody. So the scientific method tells us that we want to develop a question, go into our research to develop an overall hypothesis. It gets a little bit more complicated when we're talking about biomedical research. So when I entered my lab, I knew I wanted to work on diabetic kidney disease. And because science is the closest thing I have to magic, I decided that this lab will work. So in order for me to come up with how I want to study diabetic kidney disease, I did an initial literature review. I went into several different sources from our ultimate source called PubMed and did months and months and months of studying and looking into the different sources and what's being currently done in the field in order to find and identify small gaps that I could develop into a project. From then, I brought that project to my PI and I said, this is what I want to look at. I knew that hypertension and diabetic kidney disease go hand in hand. And I also knew that type 2 diabetes confers over 98% of all diabetes cases. So it made me think, well, what are the ways in which diabetes can affect the overall health? And then I saw some papers that suggested that lipids instead of the normal factors that you may see when you go into the doctor, like protein in the urine or high blood glucose or high blood sugars, sometimes high blood pressure, all of those things, yes, they do play a role in the development of kidney disease, but these dang lipids, these fats are contributing in some way, some way, shape, or form, but the field just doesn't know. And so I take a preclinical model of hypertension and diabetes, put that together, and see whether or not when we feed it a high-fat diet or a diet that's very similar to the standard Western diet, whether or not that would contribute to overall kidney disruption. So that was my initial question. Do these lipids actually contribute to the overall kidney disruption? And from then, we came up with our hypothesis, or our goal of the research was to identify specific processes that contribute to the intake of these fats and the processing overall of these fats in the different cell types in the kidneys. Because a fun fact, your kidney has about 10 different cell types in there, and they all do very different things. And so we wanted to look at each individual cell type to see exactly how each one processes lipids. So that was my kind of hypothesis or goal. So the scientific method will tell you, experiment, observe, analyze, you're done. A little bit more complicated than that. So once I knew what I wanted to do, I needed to prove to my PI, my principal investigator, that this would actually work. So I took some of the sources, saw that these can be reproducible because science, and I'll say this several times, has to be reproducible and statistically significant. So I needed to know, as a scientist with a huge lab available to me, whether or not this science could be reproducible and significant. So in order to determine reproducibility, I took a couple papers and redid some experiments. I saw that they used different concentrations of lipids. They put it onto these cells and saw what happened. Do the cells blow up? Do the cells stay the same? Do the cells disappear? And so I did the exact same thing. And I saw, yes, it does happen. So how do we proceed further? So I moved into a preclinical model. And from then, I developed my experiment. So all of that work just to get this one done. Next, I took and I did several different experiments with my data. I treated tiny little diabetic mice with the standard treatments and tried to see how, if they ate like a high-fat diet that kind of smelled like cheese, not gonna lie, would kind of affect their kidney health. And so from those observations, I did my analysis. Now in science, the very important thing that you want to use is called statistics. It is a mathematical model that tells you whether or not a specific comparison between two or more groups is in fact there. Okay? It gets a bit more complicated than that, but that is the essence of it. The better the statistics, the better the result, because math is king. So last but not least, you present your findings. So when it comes to me, I present each finding in a stepwise fashion. So I come up with a first group of information regarding the kidney. So I'll say, oh, so yes, the fats are affecting the kidney. That would be like the first step, right? And I would present that at conferences to other scientists. So it would be a bit more jargon, a bit more, prestige. I'd probably wear different hair. And I present. And then I publish. I have around 7 publications at this point, one of which that just got finished. I'm very proud. And thank you. And that is the way that I communicate my science to other scientists. Then that science can be moved further into the public eye by way of kind of infographics or listicles that you may see on the internet. And we'll kind of get a little bit into distinguishing those sources at a later time. So from those publications, I then delegate my science to you guys. So I'm here to answer all your questions. If you want to know a little bit more about the fascinating parts of my research, what the diet looked like, what that specific process is, ask away. I'm here for you. So that is the first part of the general workflow of biomedical science. You have your preclinical discovery, where you're in the lab, you're looking at cellular models, you're looking at animal models, you're looking at models that are not directly associated with humans, because you have your cells. Cells can be human cells, but generally speaking, you have like a general idea of how a process works. Then, once you can verify that by your statistics, you move into your clinical trials. And from those clinical trials, you do a consumption. So people's consumption. So I have personal experience with both early phase clinical trials and the consumption side because I worked in biopharmaceutical companies. So as I've stated before, my example of the scientific method was a very small scale example focused at the academic side. I'll give you a larger scale example with, the COVID vaccine, right? They take the data from about 20, 30 years ago with the SARS vaccine and saw that, these are very similar. They have a very similar structure, a very similar feel. How can we take this further? Then they looked into technology that was actually started in the preclinical discovery phase around the early 2000s of RNA vaccine technology. We've been looking into that type of drug delivery for around 21 years now. And we take a team, we build that into a vaccine. So the phases of the clinical trial, I have done a phase 1 through 3 clinical trial. I was looking at biomarkers or markers that could predict kidney disease as early as the first couple of years of diabetes. And what we found was that we had to determine that it was a safe method. And honestly, our method was the safest you can get. We just collected a bunch of people's pee and we made sure that this particular marker was in the pee or not. So it was a bit more low risk than some things that you might have heard of, but it was still a phase 1 through 3 clinical trial. So we evaluated the safety, and it takes anywhere from 1.5 years to do the whole thing. And plus, it just depends on the type of study, how many resources you have. Like if the entire world and all of the world resources are working towards one thing, it goes like this. If you don't have as many resources, it might take a little bit longer because you have to find the money and things like that, but I digress. So our phase 1, we evaluate the safety, determine a safe dosage. If you're working with the drug, you want to know whether or not a 0-point concentration or 1-point concentration is going to affect the body and how. Then we identify the side effects. Now, this goes after doing the preclinical study. Preclinical studies can sometimes include cytotoxicity or how toxic a drug can be even before you introduce it to humans. So this is kind of doing a follow-up with, okay, yes, we saw it is absolutely safe, so let's test the safety again. The next is our phase 2. Phase 2 is to test the effectiveness. How long will this work? If in our case of our biomarker study, we wanted to know how effective and how accurate the test was in predicting kidney disease in early stage diabetes. So we did about 50 or so patients, and we saw over and over and over again that it did it. It worked. Yay. Then we further evaluate safety. Do safety testing again. Safety is a common phrase within a clinical trial. Every single phase has safety testing. Even in the preclinical phase, we have safety testing. So phase 3 is confirm the effectiveness again. So once in phase 2, you tend to start with a smaller population because you want to know if it works within the most ultimate healthy individual. Then you can move into more high-risk populations. So you confirm the effectiveness, monitor the side effects, and that's when you get the, fast-talking guy at the end of the commercials, and then you compare to other treatments. So typically you have several different drugs for the same exact disease. And so you want to compare and contrast the effectiveness between each drug. Then you collect that information, and that information goes on clinicaltrials.gov. If you were ever wondering where all of that information is reported consistently, it would be there, clinicaltrials.gov. Then, and then finally we reach phase 4, which is the one that most people are familiar with, where they provide additional information over time. And after approval from the FDA, USDA, they can include more risks, benefits, and the best use. There are drugs who are still undergoing clinical trials to this day to discover even more uses for them. For instance, Humira. If anybody's heard of Humira? Yeah, Humira. So it started out as what, a rheumatoid arthritis drug? And then they were like, because it is a anti-inflammatory drug, can we apply it to other inflammatory diseases? So then it opened it up to psoriasis, it's opened it up to PCOS, it's opened it up to Crohn's disease now, just recently. So this is kind of how it goes. It's always going to be ongoing. The process of science is never over. There's never quite a finishing touch because there's always going to be a different use for a drug that you develop. And why, reinvent the wheel? So most of these clinical trials are reported on clinicaltrials.gov, and you can also find them in reputable sources. So we're going to look a little bit at the anatomy of a research paper. We have our good paper with our healthy heart and our bad paper with our not so healthy heart. So in a good paper, you want to look at the peer review. Peer review is when they take a bunch of different scientists from the exact same field from different places. I've personally gone through the peer review process, and I've had at least 4 or 5 people look at my work from my field. some people that I greatly respected. So it was kind of nerve-wracking. But they basically look at the reproducibility and the statistical significance of your science. And that can take anywhere from 2 months to a year. So the next thing you want to look at is the Digital Object Identifier, or DOI number. It is the reputable number for all reputable sources of peer-reviewed papers. Every peer-reviewed paper will have a DOI number associated with it, as well as something called a cross-reference number. You can sometimes use them interchangeably, but most of the time you'll see a DOI number that has like a really long kind of link looking like thing, and the cross-reference number, which is a very short number that goes at the top of the paper. Then you want to find something that your reputable sources show up on SciForum statistics. SciForum is a database of all the refutable sources that you could possibly find. And they sort them by impact factor, or how many times they've been cited between other people within that field. They even have webinars on different scientific topics. So it's a very good source for learning just a little bit more about different fields within science. Medline and National Library of Medicine are databases that also serve the exact same purpose, and they have a bit more they're a bit more expansive because they may include a bit more of the international journals, so you can see what's happening across the pond. Then you want to know, like, if you are looking at, let's say, NPR, for instance, and you want to know whether or not you're getting the most accurate information, I encourage each and every one of you to always click on the links within your NPR articles, because then you can see the primary sources that they derive that article from. 10 times out of 10, all different news outlets will kind of filter out the jargon and help science be more accessible. But sometimes that can lead to some misunderstandings. For example, there was a study in Singapore where they said that COVID-19 only affected you from outside. If you were outside, you're going to get it, right? And when you look at the sources a little bit more, you find that that study was actually just done on one small construction site with about 10 people. And it wasn't verified via statistics because the N was too small. So that you would only find from clicking around on the links and trying to actually look at the primary sources. And I'll talk a little bit more about the primary sources on the next slide. So again, link to reputable research articles. And then finally, author credentials. Not saying stalk people, but you can definitely Google the people who have written the articles to make sure that they actually know what they're talking about. You want to know whether or not these people are giving you safe information. And if they're not, then you can look for another author who will give you safe information. So what you don't want to find is biased websites. Sometimes we have the unfortunate habit of looking for information to confirm our beliefs instead of opposing our beliefs. So we kind of want to have a good balance. We don't want to have that confirmation bias. So you want to look for, if you're thinking, oh, the sky is blue because I say so, you don't want to look for a source that says, yes, you're correct. You also want to look for a source that says, maybe that's not right. So you want to stray away from a biased website. If you don't see a DOI or cross-reference number and they're claiming to be a scientific article, that is untrue. Every refutable scientific article, and I say this as an author of 7 papers, will have a DOI or cross-reference because you know from those numbers that they went through the process of peer review. So they are reproducible and statistically significant. And then finally, no peer review process. There's a funny saying in science that if you didn't write it down, it didn't happen. And so if there's nobody, I would extend that a little bit further to say that if the people in your field don't know about it, then it probably didn't happen. I could write hundreds and hundreds of millions of pieces of data in my notebook, and if I don't show it to my other scientists to get the verification that I'm on the right track from others within the field, then I'm just confirming my own biases, right? So if there's no peer review process, then it's safe to say that this is not a good source. So finally, we want to know the importance of primary sources. So a primary source is, for instance, an original research article. Somebody went into a lab and they did the experimentation, they made it reproducible and statistically significant, and they reported their research findings into an article with the aforementioned qualifications. Then legal documents and reports are also a really good primary source because it comes straight from the horse's mouth. You're looking at the discovery documentation that's available for something. You're looking at a specified report. So next would be our secondary sources. Secondary would be like a literature review, which is basically a summary of the entire body of research. Somebody took the time to look through hundreds of research papers, I've done that before, and write a summary of each finding. And you can either have it as in scientific speak, which, sometimes can get a little heavy on the jargon, but I do encourage to look at, or it can be kind of filtered into a more accessible language. And that's a similar idea with textbooks. Textbooks take the entirety of a field and kind of condense it into more reasonable descriptions of what the field is. So tertiary sources include encyclopedias and bibliographies. Encyclopedias being, we kind of see those, I don't know if everybody knows what an encyclopedia is. They kind of sort it by alphabet, and then they kind of talk a short, brief description of a field or a topic. And then your bibliography would be just a list of all of these different sources that kind of are about a topic. Now, when you're looking at secondary and tertiary sources, you always want to go back and fact check with the primary sources that they derive from. 9 times out of 10, a book, a textbook, an encyclopedia, literature review will have a bibliography, which is a tertiary source, or a list of all of the sources where their information came from. So that is a great way to find the primary sources associated with the information that you're taking in. So now we come to our audience participation. I'm going to encourage everybody to think like a scientist. So we're going to pretend like we're scientists who are developing therapeutics for a disease called never going to give you up-itis. It is a disorder that was discovered in the Midwest, where people who heard Rick Astley's "Never Gonna Give You Up" would begin to violently dance around and be unable to stop. So they physically cannot give you up, let you down, desert you. So I want you to describe which field of scientist you would employ. So would it be a cardiovascular scientist, neurologist, immunologist? Which field? You want to ask a question, research some literature, come up with a hypothesis, maybe some experiments, have fun with it, observe, analyze, and draw your conclusion. And then you want to kind of think about what's the most important part of the study design and what populations would you want to choose. And then finally, choose a recorder and a lead scientist who will tell me your lovely ideas. So, go. There's no wrong answer here, okay? Congratulations, you're basically a scientist. All right, and that is it for me, so I'm open for questions. Yes, question.
Could you please describe what mathematical, the phrase test that you would do your research?
Sure. Sure. One of the most common ones that I use is called the analysis of variance, or ANOVA test, ANOVA. And that basically takes several different groups with either the same treatment or different treatments and compares the, how do I put this? And compares the variance in which they would be, like the difference between the groups. So you can do it via a two-factor, so you have your treatment group versus a condition, or you can have it just based off of the treatment group. So that's one of my more popular ones that I use. It was basically like a analysis of the differences between two groups. And I would write that out for you, but I can't. Sure.
So I'm interested in the diets which affect the kidney. You're talking about what kind of foods affect the kidneys and what kind of food should we avoid so that we can have a good kidney life.
Yeah. So it's safe to say, so our Western diet or Western-based diet is about 45% fat. So you want to really look into the composition of fats when it comes to your actual diet. Everybody's heard of saturated and unsaturated fats. What we're finding is that you kind of want to stay away from dietary saturated fats, because when we treated these mice with our dietary saturated fats at a high concentration, the 45%, we saw that they had worse kidney outcomes than the ones that had a lower diet that was less than 5%.
Yeah. I read about a person who is brain dead that had a pig's kidney attached to them. And the person survived, I think, for 54 hours. And I wasn't sure why this was such a hyped up in the media, because I thought decades ago there were instances where scientists had used pig organs, sometimes to transplant into people. So I wasn't sure what I was missing.
Gotcha. Well, I'm not really familiar with the study, but my first thought would be, what are the technologies that they used in order, and how are they different from 30-ish years ago?
The kidney is some kind of enzyme that usually when you're there's a pig's kidney and somebody rejects the kidney, but they treated this kidney with a special enzyme. They found this enzyme that causes you to reject the kidney, and then the person would not reject the kidney, and measure the urine output and all that, and it was.
Oh, that's very interesting. Again, I'd have to look into it, but I will, and I can give you my email later, and I'll email you back.
Anyone else?
Yeah.
I'm just curious as to what, you personally, what made you decide to go into the particular area of research that you're in right now?
Oh, I love talking about me. So diabetes is very close to my heart. My grandmother suffered from diabetes, and she actually unfortunately passed from end-stage renal disease, which is total kidney failure. So that kind of made me very interested in how this happens. You know, how could we not have caught it earlier? What could we have done? Were there specific things that we should have done beforehand when she first got diagnosed? And so that kind of made me very passionate about that. And that's kind of what started the show for me. Oh, she was 73. Yeah. No, she never had any other, to my knowledge, she didn't have any other issues with her kidneys. It's just the simple fact that she developed diabetes, and she did have something called metabolic syndrome, which is basically the overextension of fat in your frontal area, like your frontal part of your stomach. It's called visceral and subcutaneous fat. She had a larger more pronounced stomach that kind of contributed to that condition. Yeah, what? I believe so, yes, hypertension as well. Yes.
All right, we have time for one more question.
Hi, you had mentioned some of the biomedical biomarkers. I was just curious, what were some of those?
Yeah, sure. So one of them is called KIM1, kidney injury molecule 1, and we found that it is secreted in the urine as early as first year of kidney disease. And the other one was MMP9. Those are both still in their preclinical phases, so I'm not sure if they have developed any clinical tests after that, but we have found that in preclinical models that can be as early detectors of kidney disease. Yes. Thank you.